Excellent. And tonight we have David Cappaert. He works with the Institute for Applied Ecology on pollination biology. He also works on bee taxonomy in collaboration with USGS and others on resources that might make species IDs or keys more accessible to non-specialists. So tonight, David will be presenting on pollination biology in the Willamette daisy, conservation of an endangered prairie plant. David, take it away.
Oh, wait,
Cool.
I have to stop sharing here. Sorry.
Okay. I'm trying to share. Let's see.
Stop share. There it is. Okay, now it's your turn.
Okay. So you can see my screen? Yes?
Yes.
With an insert.
Everybody else should mute because we get backlash otherwise if you.
Perfect. Thank you, David. And David, we can't hear you if you are speaking.
Oh, there you go. Okay. That helps. Okay. So yes, David Cappaert. I came out to Oregon in 2019 to work on this project as a seasonal, and I've sort of stuck around for a few years now. And at the Institute for Applied Ecology, it's mostly plant folks there. So I'm a token entomologist. But we have had these couple of projects related to pollination. One is in coordination with Susan Waters, who I think you've heard from in Washington and Olympia. We do pollination network ecology. And so we go out and collect these from every kind of plant that comes into bloom in a whole series of different prairie ecosystems. But the other big piece of what we've done in Oregon particularly is to work with this Willamette daisy, Erigeron decumbens is its proper name. And some of the folks who've been involved in this, one is Tom Kay, who's the director of the Institute for Applied Ecology. And I see that he's on this call, so he's invited to chime in anytime it makes sense, especially if I fumble any aspect of the botany. That's not my strong suit necessarily. So Tom has sort of he's been very interested in Willamette daisy, trying to get it reestablished, understanding everything that he can understand about it. And then I've worked with other IAE staff. Right now, permanent people are Christina Mitchell and Scott Harris. They're involved with this. And there's been a whole series of seasonal people over a period of time. And in the picture here at the bottom, that's Tom, who's taken a picture of Jacob. And that right there is a super healthy stand of Willamette daisy. So hold on, I gotta go forward. There we go. Okay, so from the entomologist's perspective, what I have to say about this is that in the restoration conservation world, there's of course huge efforts out there to reestablish native plants in Western Oregon prairies and all kinds of places all around the country. And the concern is usually from the plant perspective. So what does that plant need in terms of soil, hydrology, light, competition, all those sorts of basic questions about ecology. But there's one piece of this that sometimes is sort of an afterthought, that being the pollination. And so we had this unusual opportunity to be able to look at pollination of one of these endangered plants. So here's what it looks like, again, in a couple different views in the field on the left. And this is a little experiment I will refer to in a bit about we did this really fun thing where we put honeybees on sticks and used them to brush pollen from one flower to another. We had learned that from some other folks. So that's what the plant looks like. When it comes up in the field, it has an interesting presentation. I'm guessing from the plant's point of view that it's doing its best to be available to pollination for a long period of time. So it comes into bloom over a period of about 3 weeks, starting about now. And the flowers, some of them will come in early and some are 2 weeks later. and within any one particular patch of flowers, they will be in various states of phenology. So this picture on the left here, you can see a flower in the lower left that's not out at all. And like every other Asteraceae flower, they're really a set of florets. So there's going to be 100 flowers in this disc. And here's one in the bottom that is not out yet. And here's one where the outer 20% of the florets are open. And here's one where the entire flower is open. So that's how the plants come out. The asters are really interesting in that they have this thing called plunger pollination or piston pollination. And it works that in the pictures here on the left, there's tight ring of anthers here, and the stigma pushes up through, and it pushes the pollen past the anthers and bears the pollen on the stigmatic surface. So it's a little strange that way. And in this picture, you can see these little loops are about to emerge. There's one that's out. So they make these little hoops, and that's what you see in that picture there. So that's how those flowers work. And they mature over the course of about 6 days. So this would be a series of pictures of the flowers as they advance. I think it's all the same flower. Here, for some reason, advance. There it goes. Okay, so from there's a couple pieces of this I'm just going to skip over so I can get to the pretty pictures of bees and such. From the plant perspective, some questions that we asked, and I'll just give you the answers right now, were number one, does this plant, I'm having my talk, I have ERDE, that's our shorthand term for the Willamette daisy. Does it require pollination in the first place? And clearly it does. So here's just a little chart on the up here, which shows that if you, this was a little tiny experiment I did where we pollinated the plant once and got 20% seed set, pollinated twice, we got closer to 40, 3 times. I'm not sure why additional pollination events didn't increase the seed set, but if we enclosed the flower and prevented pollinators from touching it, there's interestingly not 0 seed set, but essentially, practically there isn't any. So that was one question. Yes, they do require outside pollination. And a second is, are they pollen limited in the first place? Because the whole question about who pollinates the daisy is not particularly interesting if there's a whole slew of pollinators that are out there that are supplying 100% of its needs all the time. We don't need to ask a lot of deeper questions about it. But what we did find with this sort of experiments represented down below, we took in the field an individual flower, and we would take then a bee stick, which is just, instead of using a brush, we're using a dead honeybee to transfer pollen from one flower to another, and this would be a supplement. So we're adding pollen to this flower beyond what it normally gets in the field. And the question is, if we add pollen, does that increase the seed set? If so, then the background rate of pollination is not adequate. And we did this a bunch of times at a bunch of different sites, and it turns out that sometimes, yeah, it makes a difference. So this would be a little chart that shows seed set for an open pollinated plant exposed to natural background. It set seed there, and if we supplemented it, it jumped up quite a bit. It's all over the map in terms of how often we saw this, and I'll get to some of why that might be shortly. So those are plant questions, but of course, I really want to ask insect questions. So what we sought to do was to sort of characterize the community of things that visit it. And because the Willamette daisy occurs in a bunch of different kinds of sites, we had 6 or 8 different places that we would visit. And we've done this over the course of 4 years now. So we have within, between years and between sites data. And the question is to evaluate the net contributions. We want to know, besides who's visiting, we want to know which of those visitors are actually pollinating the plants. And if we have, some kinds of answers to these questions, then we might be able to make some guesses about what would an optimal pollination landscape be. If we wanted to, if we felt like pollination was a limiting resource, What do you want to do in terms of conservation that will get the best result for the plant? So pretty much everything I'm going to tell you from here out is based on this super simple technique for trying to characterize the community of pollinators for Willamette daisy. So we established in each of our 6 to 8 sites a little path that would go from plant to plant, and we would walk and walk and walk on that path and net everything that landed on a flower. So we wore some deep trails into the landscape in doing this, and we did it at sites that are north and south of Corvallis. That's where we are here, where I am right now. But up here would be Salem, down here would be Eugene. So in this 100-mile or so range, we had sites, several around Eugene, and I'll show you some pictures of these things, and some that are north of Corvallis. And the sites look like this. So, I mean, something that must be said, a reason why I've done this for 4 years is because to be paid by somebody to sit out in lovely meadows in the springtime and watch flowers is about as good as it can get. And so these are the most, bucolic images of that sort of thing. That's the upside, is sitting in these beautiful meadows. The downside is going to Oxbow West, where they have these horrible little hummocks, and you're going to break your ankle anytime you hike through there. But in any case, the intention was to distribute geographically the kinds of sites we're looking at. But for the most part, they're fairly similar. They're restored prairies. They have pretty rich flora because they were established for that reason. And that included putting the Willamette daisy into those sites. And so I'm at this point just going to go through a bunch of pictures of bugs that we saw. And this list would be the kinds of taxa you would see almost any place you're going to look. We had representatives of, here's the bees. So halictids are really big. We saw some Megachilidae, Andrenidae, an early season bee, and they're definitely part of the scene for Willamette daisy. And then I'll just show you pictures of these various family Apidae. And I'm going to say a fair amount about syrphids, even though this is a bee group. If you're an entomologist, you can't ignore the syrphids when they're in your face the way they are in this particular study. So a point that is obviously well understood by all of you is bees are not just one thing. So this would be my best effort at explaining why you don't just say bees and know what you're talking about. So the little one is a Hylaeus and the big one is a Eucera. So just sort of working through these in sequence, Andrena, it's been mentioned some already, early season bees, solitary ground nesting. And very interesting thing about Andrena is that they're super diverse. In the Discover Life keys, there's 140 species listed for just Oregon. And there's, like probably closer to 600 in the United States. So they're really interesting and a challenge certainly to ID, and I will come back to that. A feature of Andrena, probably a lot of you know this, are the facial fovea. So that would be absolutely diagnostic to have scopi that look like this. There's other bees that have scopi, but this kind of flattened hairs between the eye and the antennal base, that's an Andrena thing. Sweat bees are, well, you'll see shortly, those are the most abundant taxa amongst the bees that we see. Also solitary or social, also hyperdiverse. I think Link probably knows more about this than anybody else. The number of Lasioglossum species that we have in Oregon, it's a huge number, and there is no doubt that there are undescribed species amongst the Lasioglossums. So here are just a few specific sweat bees. We usually see these two genera. So it's either Halictus or Lasioglossum. Halictus are fairly easy because there's just not very many species of these. For those of you who don't know, they're defined by having hair bands that are apical to the segment. So there's the segment, and at the end of that, there's a fringe of hairs. And so that makes something that could be either a Halictus or a Lasioglossum, that's a Halictus. And there's big ones and there's little ones. So the commonest big one is this one here, Helictis ligatus. And if you're not really good at bee taxonomy, it's really nice to know about ligatus, because all you need to see is the little tooth here, the genal tooth at the corner of its head, and you've identified it. These look the same size on the screen, but in fact, this bee here, the ligatus, is, you know, a medium size, 12 millimeters or something like that. And this one is a very small bee, and they're characterized by this metallic integument. So we just have pretty much these 4 species that we see of Helictis. By contrast, the Lasioglossum, which superficially look alike, at least some of them do, are hyperdiverse, as I was mentioning. So this is all the diversity is with the Lasioglossum. And these are sort of 2 categories of Lasioglossum. One on the right is one of the not very many species. We probably have 5 or 6 that are pretty big. And so we can identify those pretty reliably. But then there's a slew of these little tiny guys, like the one on the left here. They're often 6 millimeters or so, or even less than that. There's a whole set that are metallic. They're famously hard to identify. There's very few resources for doing a good, you know, for making that easy. And we certainly have, I would say, in our collection, 15 different taxa, most of which we don't have names for. And then here's a picture that of a bee that Link gave me, because I think he wanted to have a really flashy osmia picture. This is sort of the extreme of beauty for osmia, but they're all gorgeous. They're all have metallic hints to them, and there's, you know, green and gold and blue like this one. And these ones nest in cavities. You see them in bee hotels. You know, that's a popular place for these bees to hang out. Also really diverse. And resources are thin. It's a little hard to figure them out. You kind of have to talk to people like Link to get the true answer for these. And then leaf cutters is the other category. I didn't mention, for people who aren't like totally savvy about all this stuff, mason bees are in the Megachilidae family, and they have these scopi that are underneath their abdomen. So that's a feature of Megachilidae. And Megachilidae, the genus, also has that feature. So those hairs are where they will collect the pollen into. Called leaf cutters because they make these like really perfectly circular cuts out of leaves that they used in building their nests. And then that's just a few pictures of them in the field. They're pretty darn cute. Coleoxys, this one is also a megachilid, and despite what I just told you before, where they have these abdominal scopi, Coleoxys does not. It's naked underneath, and that's because, like a couple other bees I'm going to show you in a minute, it's a cuckoo bee, so it's a kleptoparasite, and this one is going to probably parasitize other Megachilidae. Other cuckoo bees are the in the family Apidae are Nomata. I've seen a lot of these this year at some of our sites. And one of the really fun things, I suppose you could think of it as fun, interesting things to consider is when you see a bunch of these, you're looking at an animal that survived on a meal of other bees. Maybe for these guys, it would be andrinas. So it's one of the possibilities when you see changes in population levels of bees that you expect to see. Maybe there's something going on with these kleptoparasites that are reducing their populations. And then there's the tiny bees. We've already seen some pictures of Ceratina, and we see those pictures a lot because they're hyper abundant. It's pretty variable from year to year, as you'll see in a minute, but this is, as I understand, in Oregon, this is probably the most collected bee in the Oregon Bee Atlas history. They're all over the place, and Link suggested this is related to Himalayan blackberry, which provides nesting substrates for these. That's certainly a thing. And then we didn't see a lot of these, but another little tiny bee that you'll see is the masked bee Hylaeus. And then occasionally you're lucky to see these really big, beautiful, hairy bees. There's Eucera earlier in the season and later Melisodes. They have these great big pantaloon-like scopi that they carry the pollen around on. This one does not have a big scopa for carrying pollen because it's a male. These are the longhorn bees, and the males have these crazy long antennae, which they use in mating, which is pretty interesting. Okay, so past the bee part, now I have to talk about syrphids a little bit. Syrphids are super abundant. You'll see in a minute. They feed on nectar and pollen, unlike some other flies that only eat pollen. These guys can also drink nectar. After bees, generally, if you sort of read on the internet about pollinators, they're cited as, after bees, the most important group that does pollination. They're kind of cool because they're predaceous as larvae. Some of them are also aquatic as larvae. And here's a few pictures of them. Probably generally the most abundant are Toxomeris. There's 2 different species of those, which are really easy to differentiate. These are really small. They're 6 millimeters or something like that. And, it takes a while to get used to, but they're as beautiful as bees are, if you ask me. But I'm an entomologist after all. And I mentioned they're predaceous as larvae. So it's kind of a fun thing. The first time you see in an aphid colony, you see one of these syrphid larvae, you're sure it is a caterpillar. They kind of look that way. But then you see these little teeth on the end that they pierce and suck out the life from these little aphids, and it's a syrphid larva. And then not all syrphids are created equal. So the ones I just showed you, they're very small and they're not particularly hairy. The Aristolis, the drone flies, are basically bee mimics, and not just in terms of what they look like, but also in terms of having these really hairy bodies that are probably pretty efficient at carrying pollen around. And some of these are aquatic as larvae. And here's another couple little species, Platychirus and Sphaerophoria. Platychirus are called sedge sitters, and I forget if there's a common name for the Sphaerophoria. And then I'm getting into a few of the sort of miscellaneous other stuff that we see. These aren't really going to be very important. They're just fun to put pictures in of. Tachinids are nice because if you see a big fly with a spiky butt like this, that's characteristic for tachinids. And they are parasites, but not on bees. And then the bee flies, they're kind of fill some of the same role as the syrphids do. Can be super abundant. This year I've seen, at one of the sites I frequent, I've seen more bombylius than anything else by a factor of 2 on certain flowers, on camas, for example, on popcorn flower. Absolutely the dominant visitors. These are also parasites on bees. So when you see a lot of these, you have to wonder. And then we see beetles. One that's really interesting to me is the cucumber beetle, which I think everybody disses, including me, because they're agricultural pests. And so they seem like not very exotic or very interesting, but I see them on everything. They're very generalist. I see them early in the year, so maybe they're important on some plants. Don't know. We don't see a lot of them on daisies, so they're not going to be a factor here. This one up here is a blister beetle. And blister beetles are interesting because they're another natural enemy of bees. And you will sometimes find on a pin specimen, you'll find these little dried up larvae. They're called triangulans. It's the life stage of the meloid beetle, the blister beetle, that attaches to a bee and gets carried around. So you see these on Andrena and on Halictids, probably other taxa too. And this is, purely et cetera stuff. These are unusual to see, but that's one of the benefits of sitting in, patches of flowers and watching bees for hours and hours and hours. You see rare things that are pretty interesting. This picture on the left is a earwig, and I would never have thought of an earwig as being associated with flowers, but this one is obviously eating pollen. It's what is this plant? Can't remember for sure. It could be a Sidalcia. An occasional moth of some kind. It's sort of a peeve of mine that here's a butterfly on a flower, and they're often regarded as pollinators, but certainly in the systems we work in, they're completely irrelevant. They're really very uncommon to find on flowers. And you also notice that they have these long legs and a tongue that reaches down. There's not much opportunity for pollen to be moved around from one flower to another. And then I've deliberately put the honeybee in the lower right corner here because they are also not very significant. As much as they capture everybody's attention, when you talk about bees, they think honeybees, but in fact, except for sites we have that are close to ag, where there's a whole bunch of bees that have been maintained in colonies, in natural settings, we just don't see them very much. Okay, so that's the easy part. I've told you a little bit about, I've showed you the pictures of them, and the easy part that's coming up is I'll tell you about the abundance of those that we've seen. So here's some numbers. So when we do this thing running around and collecting, and this is a composite for a whole bunch of different sites and a couple of different years, black bars in this chart are flies and red is bees. So you can see in terms of sheer numbers, in the top 6, they're all flies. Taxomeris, the little guy, this is also Sphaerophoria, another small syrphid, Platychirus, another small syrphid, various other Diptera, Eupiodes is another small syrphid, and then Halictus. So I've got Halictids in the second position, and then way down here, the Lasioglossum and the Halictus species. So it's a picture that's pretty dominated by flies in aggregate. So we're going to have to think about flies. But here's something that's super interesting. If we look at the heterogeneity of this, it's like astonishing how variable things are. So to take one extreme example, if you wanted to understand the pollinators of Willamette daisy, you could go to a patch of flowers and you could watch it in 2019, and you could make charts like this, and you could start to talk about what's happening. But that won't work, both because of variation from one site to another and variation from one year to another. It's huge. And this is generally true for invertebrates, for people who study them in forests or in turf grass or whatever. It's amazing how much populations of basic members of a community go zinging up and down. So this chart shows you 3 different sites. I just picked arbitrarily 3 different sites. And compare Taxomeris right here. That's one of these small syrphids. At Finley, which is the orange bar, it was like crazy abundant. It was, far and away the most common thing we collected at that site. At the other 2 sites, which are also prairies in the Willamette Valley with similar sets of plants, they're virtually absent, right? Lasioglossum is relatively well represented at each of these 3 sites, but here's another fly, and it's pretty different from one site to the next. And like even for, if you want to just consider bees, these 2 at the end, Andrena is really common at Ankeny Refuge, and much less at Finley, and scarcer still at the site that we call Kingston. Ceratina, same thing, really abundant at Finley, substantially lower at Ankeny, and then virtually absent at this other site that's called Kingston. So the bottom line here is you don't know what's going on with the population of pollinators for a particular plant unless you look at a lot of places where that plant might occur. Okay. Keep having to look for my arrow here. So now let's look at variation from one year to the next. And this is also really messy. So I'm comparing 2021 and 2020. And between those 2 years, and I'm combining three different sites together. So between those 2 years, Lasioglossum is pretty well represented. Taxomeris, one of those little syrphids, was like nothing happening in 2021. It was hyperabundant in 2020. Halictus, pretty substantial different from one year to the next. Andrena, I'm seeing that this year too. I'm seeing way less Andrena, but these are very different numbers. And this is average for a number of sites. So overall, in the Willamette Valley, I'm going to say there's something going on between 2020 and 2021 that has a big effect on Andrenas, has a big effect on Platychirus, has a huge effect on Taxomeris. So bottom line here again is it's just you've got to know a number of years running before you have sort of a sense of what may be happening. I also noted that in looking, and we collected a lot of insects, right? Because we're looking at hours and hours of sampling from a set of plants at 6 or 8 sites. And out of these 2 years, 2021 and 2020, only in one of the years did we see these relatively rare things, Melisodes, Escherichia, Eucera, and misspelled there, Hippolytus. So anyway, we do have some sense of what we're going to see that shows up to ostensibly pollinate Willamette daisy. But we don't really know anything about the efficacy. And this is everything that you see in the world of pollination ecology, the papers that are published. This is one of the things that really kind of gets danced around a bit. I mean, there is serious effort to try to understand this, but a lot of times the pollination story is told in terms of the visitation frequency. So if I told you just a minute ago that there were Toxomeris, this little fly, was like enormously abundant because we have charts that show how often it can visit the plant, is it really carrying any pollen? Because the only thing the plant cares about is whether pollen gets from one anther to a stigma. That's when pollination is affected. And just to take a really extreme example here, here's 2 lasioblossoms. This one is Taitusi, and you can see she's doing a pretty good job of carting pollen from one flower to another. And here's another lasioblossom, a little one, a Dialectus, that looks pretty denuded of pollen. So maybe it's nectaring. I don't know, but to call the visit of one of these as equal to the visit of another one is not telling very much of what's going on. So beyond abundance and phenology, 2 other pieces that you want to try to understand are the morphology of the insect, how well it can, what is its capacity to carry pollen, and also its behavior. How does it interact with the flower? And does that effect transfer from anther to stigma or not? So here's one take on that. So I'm going to consider the morphology piece of this. So here would be a hypothetical, I just made this up. Okay, so this is a hypothetical pollination network. And the way it works is I've put pollinators up here. And so here's Halictus ligatus, the sweat bee. Here's a different sweat bee. Here's Ceratina, the bee we've talked about a bit. Here's one of these syrphids. And down here at the bottom are plants that they visit. And the width of these lines that connect Halictus ligatus to this plant, Potentilla, tells you how much visitation there is. So Halictus visits Potentilla quite a bit, and Toxomeris visits all of these different things quite a bit. So we've got a visitation network that would suggest that this is a pretty robust system, that there's a lot of redundancy, that everybody's sharing in the wealth of the pollination. But if you look at some of these example insects, here are 2 pictures of insects that are quite abundant in the mid-season in the Willamette Valley prairies. On the left, here's Toxomeris, which I keep talking about. And if you look at it really carefully and count the individual pollen grains, there are 11 on this side of this insect. If you look at a Halictus ligatus and just count these small areas of its body, there's 62 grains of pollen there, 60 there, 70 there, 120 here, and I haven't begun to map the entire body of this insect. So its capacity is enormously greater. So can we factor that in when we try to understand like where pollination is occurring as opposed to visitation? So as this thought experiment, I'm going to take that chart I showed you before, the hypothetical pollination network, and I'm going to adjust it by pollen load. And these are sort of made up numbers, but they're not too far off of what's probably going on. So I decided that for a little unhairy, very small Syrphid Fabrifolia, I'm going to give it a pollen load rate of 10. And same for this other really small one. In the lower left corner here, this Aristolis dronefly, which is a bee mimic, they're really hairy and they probably carry a substantial load of pollen, way, way more than these other Syrphids, and so on. So I put numbers based on the relative hairiness, sort of what I've seen in terms of you know, how well they seem to be loaded. It's going to be affected by their behaviors. So for example, a nomada, which is a parasite, is not designed for collecting lots of pollen because they're parasites in the nest of other bees. So you don't expect them to be carrying very much pollen, and so on. So I put these numbers on here, and then I reran this network. And so down below is the network that makes the assumption that a visit is what counts. And up here is based on the assumption that the amount of pollen that an individual insect can carry is what determines the efficacy of this network. And it's a completely different deal, right? So all of these, I have to move this stuff out of the way. All of these little syrphids, Sphaerophoria, Toxomeris, they're barely connected to anything. These plants at the bottom that have connections to those small syrphids are really not benefiting at all. So I could walk through these individual examples, but you can see that it's a very different picture, and it changes every assumption you make about what's going on. So there's a lot that could be said about how you sort of deal with this. And I'm not going to go into it unless there's questions later about this, but this is something that everybody who collects insects, including just ordinary folks who are out there, you know, you all who are just trying to add to your list of cool things that you've seen, it's worth thinking about the extent to which visitation equals pollination. Clearly, it doesn't always do that. So there's that. Now, another variable here is going to be behavior. So what the insect is.
You're okay. Let's see. Lost your volume, David.
Okay, I'm back, right?
Gotcha. Yep.
Yeah. Okay. Too many buttons I'm pushing. Okay. So I hate video. It's really time consuming. I heard someone else mentioning this. It's just, I can go out and in 30 minutes, I can take 12 awesome photographs and, you know, I'm satisfied with them. Video, you spend hours and hours and it's really frustrating because of the focal issues. You know, when you're macro, it's really hard. But sometimes I do video just because you can learn a lot by doing it. And if you want to do video of bees, a really good flower to do is sunflower because it's all in one focal plane and it's a great big flower, so you can see a lot going on. So I want to show you a little piece of this. And the point is to look at how the behavior of the insects involved might affect the relative efficacy of their transfer of pollen. So here it goes. Okay, so there's your sunflower, and it's got, we're going to see halictid, melisodes, and honeybees. So here's halictus. That's a minute pirate bug that just dodged into the flowers there. So is it eating pollen or nectar or both? It's pretty clearly eating only pollen. It's not reaching down into flowers for nectar at all. So the thing to wonder about is, from the plant's point of view, how effectively is it going to move pollen from one floret to another one? Pretty effectively, I would say. And another thing to think about when you watch these bees close up and when you slow them down a little bit is the amount of grooming that occurs during the time that they're foraging. So you might wonder whether pollen that ends up on the back or the head or the wings of the insect is going to ever come into play as far as the plant is concerned. It will, because she's going to be scraping the top of her head and moving from front leg to middle leg to hind leg, packing into her scopy. So all this behavior stuff is going to be important from the point of view of the flower. Okay, so Melissodes, I'm going to just give you a second of the male because they're not very interesting. Purely nectar. He's dodging his head down in there. Males don't collect pollen. There's no point to it. Female. Now comparing to the Halictus, she is pretty obviously both collecting nectar and pollen. So what are the implications for the flower that she's nectaring? It's going to affect how deeply she pushes her way down in there. Look at all that grooming. And every time she's grooming, that involves an opportunity for pollen either to move away from the plant. She's going to pack it into her scopy and now it's not available to the plant, or she's going to be exposing it to additional stigmas. So besides being really fun to look at, this is sort of one of the things that doesn't really get thought about a hell of a lot. Oh yeah, and then I have a honeybee here. And interestingly, the honeybee is not bothering with pollen at all. It's purely nectaring. So you would see these three bees, these species we've seen on the flower, and make an assumption about the efficacy of the time they're spending on the flower. And the honeybee is probably not doing a very good job here. She's not picking up very much pollen at all. She's just sort of sucking out the nectar. So there's that. Okay, so back to my other. Where'd it go?
And there are,
Oh,
you
there
know,
it
a few questions, David, coming into the chat. I just haven't told people they can interrupt you or anything like that.
Well,
So if
that's
you can-
fine. Kate, why don't you tell me if it's
Sure, sure.
helpful to interrupt, because I'm glad to do it.
Well, okay, so let's go with David Jennings' last question. Do flies
Yeah.
exhibit consistency behavior, going to different individual flowers of the same plant species repeatedly?
Yeah, that's a really important question. And it's another piece I'm not addressing here, but the constancy of an insect who's visiting flowers is another huge variable. So if a fly goes from plant A to B to C to D and then back to A again, it's not carrying much of A's pollen by the time it has made that circuit from another set of flowers. So a generalist might be less effective for the pollination of a particular plant. In particular, if we're talking about the Willamette daisy, for example, which is not a very preferred plant. So there's a really good chance that some of the things that visit it, including some of the flies that I've mentioned here, will hit a Willamette daisy flower. And then it's in a sea of potentilla, of cinquefoil, which is a really good nectar plant. And it will visit the cinquefoil, it will visit an oxeye daisy, it will visit an Oregon sunshine flower, and then it'll come back to the Willamette daisy. And it's not going to be doing a very good job there. So David is right to think about how the circuit that an individual pollinator follows might affect its transport from one plant to the next of the same species. Flies are, it's kind of all over the map, just like it would be with bees. So there's some that are quite specialized, but generally speaking, I would say that the syrphid flies that we see in our system are pretty generalized. So they're not going to be highly faithful to one particular species as they move around. But like everything else, of course, that has to be caveated because the insect is making choices about flowers based on its nutritional needs, based on availability, based on what the flower is doing at any particular point in the day, because it produces more and less nectar, more and less pollen at different times of the day. So there's lots of stuff involved, which is just my way of trying to avoid David's really good question with a clear answer. I don't have one. Okay, anything else?
Well, once we opened that up, a few questions did start to come in. I hesitate to go backwards, but I will bring those up if we don't get to them. But then Tony Dickey asks, do Halictus females pursue nectar at any time?
Yes, they certainly do.
Okay.
In fact, I think it's fair to say that every bee that's not a parasite has to collect both pollen and nectar, right? Because it's essential to the nutrition of their larvae.
Well, makes sense. Of course. Yes, it makes sense. Good question, Tony.
Yeah.
And then Karen Wright asks, how many other asters bloom at the same time?
A bunch.
Are any of the Andrena species specialists on aster?
Yes, there are andrenas that are specialized in aster. I don't know very much about that, but I know that that is true. For this particular project, for the Willamette daisy, we don't see a lot of andrena on it. It's partly because of the time in the season. So I can't generalize and say, but I would say that the andrenas that we do see are probably not specialists on asters. But I don't, my depth of knowledge is not very big there. So any questions would be great. I want to do one other little piece before I do that. And this is a more general thing. It's beyond the Willamette daisy. And it relates to what, like, personally I'm interested in doing. So everybody in the Washington Native Bee Society and everybody that I work with needs to put names on the species on the specimens that they collect. You want to know what they are. And here's my scattershot notion of how the landscape of bee taxonomy works. So on the one hand, on my left side here, there's a really rich taxonomic literature. These bees have been studied and they've been categorized, for hundreds of years. And there's really good information about, there's much more to be done, but we have names for, like I mentioned, 140 species of Andrena in Oregon. They have names. Someone has figured out why they should be called whatever they are. That rich taxonomic literature, it runs into these problems of paywalls and jargon. So you can't get a lot of the papers that you would want to get to be able to do your own taxonomy unless you're willing to, either you work for an institution or you have some way of getting a hold of these papers. But even bigger than that is the jargon. And it's kind of unavoidable because the people who do the taxonomy, they require a very complicated set of terminology because it's just necessary to understand the slight differences. So you need to talk about rugulose versus rugose versus striate versus et cetera, et cetera. You've probably all seen a lot of these terms. But for those of us who want to identify bees, sorry, boy, I really jumped. For many of us that want to identify our bees, we can't work with that jargon very easily. We need to have some degree of translation. On this one hand, there are legitimate specialists. There's people who are really good at doing bee taxonomy. Try to get a hold of them. It's often impossible. There's too few of those people and they are too busy. So if I want to identify all my nomata, and Link best help me understand this, I can find a specialist I can send them off to, and it will take her 2 years to be able to get to them because she's got boxes up to the ceiling that are waiting for her attention because there's just not that many people who understand that particular group. So that's a barrier. There's too few people to help us. There are also enormous curated collections in museums, and we don't really have easy access to those. You could make an appointment to go to Oregon State University and see their collection, but it's a cumbersome process, and they're not set up to have lots of ordinary folks rummaging through and looking at bees. So that's a problem. And for these people who have the literature, who have the specialization, who have the curated collections, they can put a proper name on a specimen. They can say it's Andrena chlorogaster. Great. Here's everybody else. So Washington Native Bee Society, grad students all over the country, ecologists who are not entomologists, land managers, citizen scientists, iNaturalists. This is a huge number of audiences that want to have a name for the things that they find. And sometimes it's idle curiosity, but often it's because they really want to understand the ecology of the system that they're working in. And these folks, without access to the literature, to the specialists, to the collections, are often going to end up having to say it's Andrena species. And for practical purposes, that is not useful because, I mean, it's useful from the citizen science point of view. It's fun to know that you have an Andrena, you have a Melisodes or whatever. But if you're trying to understand the system from an ecological perspective, Andrena doesn't mean very much. It could be, there's a million kinds of Andrena with completely different life histories and phenologies and behaviors, et cetera. So you really want to be able to get past all of this and figure out how more people can have more access. So here, I'm going to get out of this. And I will show you my attempt to get at this. So window. I'm going to end up with the wrong screen. Let me try here again. Hold on. Oh yeah, here we go. Okay. So I'm going to walk you through just very briefly. I don't have much time to do this, but through one of my attempts to try to like bridge this gap between the hyper specialists and the people who need to understand what they're collecting. And I'm partly pitching this because, you know, I just think it's really important, but also because I want folks in the Washington Native Bee Society or anywhere to understand that we can get into this. We can end up finding names for things if we want to try hard, even if we don't have an association with some fancy institution and we haven't been grad students in a taxonomy lab. I want to believe that we can make this taxonomic information more available. And so I'm going to show you this key that I built. And I will tell you that if you said, I want to build a key to Halictus species in Washington State, I would be delighted to show you how to do it. And I think citizen scientists need to have more investment in this kind of thing. It's possible. So this little example I have here, this is the Andrena subgenera of the Pacific Northwest. I'm not going to explain to you too much about how I put it together, but I will tell you that if you want to do Andrena, the reason why I picked Andrena, if you want to identify your species of Andrena, you must know the subgenus because all of the literature is based on that hierarchy. So if you have Calendrena, you need to be able to go to the paper that was written by LaBerge or one of these people who have a mountain of fantastic information about Andrena. They've studied the daylights out of it. They know what they're talking about. And you will only have access to what they have to say about it if you know what the subgenus is. So this is my effort to help some ordinary person get subgenus. So this is an interactive key, not a dichotomous key, right? So you get to choose whatever characters you want to pick. So I'm going to pick, for example, right here, apical hairbands edge to edge, okay, just like a halictid will have. And when I do that, I cut my choices in half. And if I say it's metallic, its abdomen is metallic, I've now cut this to 2 that are total matches for that. So that was pretty damn easy, right? So I have a sense that, you know, it's very unlikely to be a Tylandrena that doesn't match either character. It won't have complete apical hairbands and it won't be metallic ever. So I'm partway there. And maybe I just picked 2 super lucky characters. So sometimes it won't be as helpful. Phobia color, dark, cuts it in half. It's another way I can cut these in half. So you can very quickly, in an interactive key like this, rather than grinding through a dichotomous key, you can quickly play around with it and just see where these positive ranks and negative ranks for a bunch of features work out. And I can add as many of these as I want to. And basically, I'm developing a hypothesis for what my taxon is. So I've got, with a bunch of characters, 4 characters now selected, I have an equal chance of it being any of these here at the top of the list. But I can reject a bunch of others. So that's really nice. So let's say I do that. The other thing this key does is it will give you pictures. And this is critical. This one, not so much. It's not hard to understand, you know, apical hairbands. But I think for anybody who's trying to work their way through a key, to see pictures that look kind of like what you see through a microscope are going to be super helpful. And in that case, not so much. This might be useful to some folks, you know, because it's a little hard sometimes to see where the antennal segments, you know, where the indentations that define them are. It might be hard for some people to understand that you've got a scape, you know, and the flagella are here. And so if you see a picture, it's going to help you quite a bit to make these discriminations. And so that's my intention, is to make here is a midline that's impunctate. This is the clypeus, sort of the front of the face of an Andrena pruni. And when you get into these little details, it's pretty clear like how these things are different. Here's one of the kinds of things that taxonomists love to do is to measure things against each other. So here's an lateral ocellus at the top of the head. And the question is whether the fovea, which is the set of flattened hairs to the inside of the eye, whether it's more or less or equal to 1 diameter. And so there would be a picture where it's about 1. Here would be a case where it's more than 1. Here would be a case where it's very close, the ocellus and the fovea, and so on. So this picture stuff has got to be part of it. This is a super important feature of Andrenas, the labral process. So here's the clypeus, the bottom clypeus. So we're looking underneath its face. And they have these little projections that are really rich for details. So this one is immediately recognizable as Barba labris because they have this strap-like labral process. So that's helpful. But the other options are, it could be long or short. You could have a little indentation or not. Here's a short one that's rounded at the end. It could be bidentate with teeth in it, and so on. So here's the other features of this thing. So let's say in my original example, I said that it had complete hair bands and that it had a metallic integument. So these are my two top, Euandrina and Scaphandrina. So if I go to a side-by-side comparison and I select Euandrina and Scaphandrina, now I can see the entire set of characters. There's 44, I think, in here. So you can see either where there's overlap or where a red bar here means that for the trochanteral flocculus, which I know you'd have to see some pictures, probably most of you to know what the heck we're talking about. But if you could look at that and understand what it was, and I have a picture of both of these, if it has a complete trochanteral flocculus, that's a Scaphandrina thing, and you will not see it in Euandrina. So you can get as much evidence as you want about how close this match truly is. And then another thing you can do is for any individual one genus or subgenus, there's a page here. And if we chose Euandrina, here's all the text from the original taxonomic work on Euandrina. A lot of this work is done by this guy Laberge and various students of his. Laberge is really interesting. If you read one of these original papers, it will start out by saying, in the exploration of Euandrina, we looked at 8,475 specimens. It's like, oh my God, it's astonishing. These people are giants who do this work over the course of decades. And so here's what he said, and this is, even though it's back in 75, so far as I know, there's not anybody who's like updated this in a very substantial way. This is the subgenus description. This is what everybody else uses. And this all comes from the primary literature. I just quoted it. But this is probably the stuff that you would want to see if you really want to get into the nitty-gritty about whether what you have is Euandrina. And then there's pictures of many different Euandrinas from various sources. And one of the funnest sources is one of these specimens from Oregon State is determined by Laberge. So I'm going to say that if he said this is Aracoma, he's the Bible on this. So that's a really good ID. If it says David Cappaert determined this, you might want to take a second look and make sure that he knows what he's talking about. Although I'm confident about this one. By the way, this one is one of my favorite, Andrena astragali. And one of the reasons I really like it is that if you collect an Andrena from a death camas flower, it is going to be astragali. It's a very tight relationship between those things. So anyway, I put in a whole bunch of pictures from various sources. So you can look at the, here's the metallic integument of one. You can see when you zoom in, sometimes you can see whether the hairs are simple or plumose. You could look sometimes at the details of the wing venation and so on. So this is my, magnum opus of this month anyway. And as I say, I mean, I promote this partly because I think this is a game changer. Your alternative in working through a paper dichotomous key that doesn't have photographs is going to be really frustrating unless you can spend a month of your time trying to just sort all this stuff out. So I will leave it there for now. And any questions or discussion, I'd be delighted.
Wow, excellent, David. And I would love it if you had a chance, while we're asking some questions, to pop in a couple of links into the chat, if you can, to some of the references that you've been popping up there, because I think entomologists have surpassed botanists in a way, and that's maybe a big statement, I'm not sure, but we are using keys. Right now working on some species back in the down in Nevada in the Intermountain Flora, and they are all hard copy, outdated, etc. So I am loving the key work that you are doing and inspiring folks to be able to do some work on their own. So thank you very much. And
yeah, and I put the link to that one in there, and I'm playing with some other keys, but you know, I can't stress enough. I mean, coming into this field, I'm an entomologist. I'm not a bee person, right? I mean, I've got a whole career based on forest entomology.
Yeah.
And I'm going to say to other people who are like I am, and especially like I once was, there's this huge intimidation. Like, who am I to try to put a name on this thing, right? This is for people who, this is for experts, and ordinary folks can't do this. And I want to say that a lot of times that's actually correct. Ordinary folks can't do it. I think it's going to be very hard to name your dialectic species without getting to the nitty gritty with Jason Gibbs's key. It's not going to be easy. And so the experts who would say, you know, don't tread too easily into this space. It is hard, and that's true. But I think we need to find the places within taxonomy where it can be fairly straightforward and take people as far as we can get them. And so this is what I'm trying to do with the Andrina subgenus thing is, I don't know how to identify a particular Andrina species other than going to the literature at some point. You have to do that. But I think I can get you to subgenus, and it's way more fun to say I have an Andrina, and it's a microandrina, and I think it's chlorogaster. That's way more fun than saying I have Andrina morphospecies number 19.
thank you.